So this is mainly for the people now listening at home. For those people that haven't watched those videos, you should go out and watch them because it does kind of do a nice overview of everything. There are some kind of factual mistakes made within it, and we'll address some of those as we go through. Not a huge issue. You get the general overall idea from both videos, which is kind of nice. So your replication process begins with a primer. Okay? And then that primer then has two ends on it. You have the 3' alcohol and your 5' phosphate group, if you remember back to your general structure of your nucleotides. The phosphate group ends up acting as the electrophile, okay? Because that phosphorus within your phosphate group is extremely positive. So that's that phosphate. Why might that phosphorus be extremely positive? What's it connected to? It's connected to a whole bunch of oxygens. Those oxygens all withdraw electron density, right, causing that phosphorus to be very, very positive, which then facilitates or allows for the 3' alcohol to come in and attack the phosphorus and kick out the diphosphate. This is a favorable interaction similar to what we saw for our conversion from ATP to ADP. You're kicking out that phosphate group, which now has more resonance and has more hydrogen bonding interactions. You're starting at a very high energy nucleotide, which now facilitates that bond transfer. Okay? So that's why we need our triphosphate. Okay? We have to release that energy somehow, or account for that energy somehow. As far as our direction for DNA synthesis, we synthesize, why is that arrow shown that way? In our 3' to 5' direction. Right? DNA can only be synthesized in that direction, which becomes a bit of a challenge. So one of our strands, it's not an issue, because it is oriented in our 3' to 5' direction. But what's the issue with the other strand? It's now the opposite direction. Since our synthesis can only occur in one direction, we end up forming these odd loops. Okay? Those loops are to now facilitate the synthesis in the opposite direction. And there are a couple theories upon how that synthesis occurs. So in the videos you saw, it was individual nucleotides get inserted in. Turns out the individual nucleotides theory doesn't work. Okay? And what we get instead are these large sections of DNA being synthesized out. Those sections are referred to as Okazaki fragments. Okazaki fragments because?
They're not attached to one another.
So they're fragments, so they aren't attached to each other, which then means we need some post-replication processing, because we're now going to have to link those to each other. Okay? So we don't have the phosphate backbone all the way through that. But why the Okazaki? It was Okazaki's research group that was the one that actually discovered this and proved this to be true. Enzymes that need to go through and do this, we've got a lot of them. The primary one that we're going to, or that you hear reference to, is your polymerases. And there's all sorts of different types, okay, based on what their specific role and when they were discovered. Okay, so DNA polymerase 1, typically responsible for repair and patching. Polymerase 2 is your synthesis of the new strands. Okay, interestingly enough, that one was probably discovered second, not first. And then we got a whole bunch of, a slew of others, which is probably supposed to be polymerase 3 there, 4 and 5, which are responsible for proofreading and repair. Okay. Probably the most important aspect that you'd see on tests is in reference to that Okazaki fragment. Okay. These are roughly, depending on the class, be it prokaryotes or eukaryotes, I think it's anywhere from 100 to 200 nucleotides versus 1,000 nucleotides. I forget which class each of those are. Those get kind of inserted and stabilized on your lagging strand.
I think for polymerase 2, polymerase's new strand, you're talking about like creating the new strand?
Yep. So if we're looking at this polymerase in that drawing, that polymerase is likely DNA polymerase 2, because it's the one synthesizing the strands, the new strands coming out of this. So they do have to match the pattern because they have to pair up. If they don't pair up, you don't get proper DNA synthesis. So that is a whole nother story and a whole nother question that I would personally like answered. How does it know that those fragments need match? So you got 100 to 200 nucleotides that need to be pre-built to be put into the DNA. How does it know the sequence of those nucleotides? Okay. One possible argument that I could potentially see out of that is it doesn't. It's just a random sequence of nucleotides that happens to best fit and match. You then have a repair mechanism that comes through, and anything that mismatches fixes that mismatch. Okay. I don't know if that's true or not. Okay. But I do see that as kind of an odd complication of that. Ooh, there we go. So we put it all together and look at all the individual units within it. You've got a nice kind of figure that goes through and shows all of the possible combinations. You've got a gyrase at the very front. That gyrase is part of the supercoiling, so it's undoing that supercoil. You've got your helicase. So your helicase is now undoing just the double helix and unzipping it at the same time. As soon as you unzip it, you now have single-stranded DNA. Well, the whole issue, or the reason why we had double-stranded DNA was to do what? To protect the DNA so it doesn't get affected. In this whole process, what do we end up doing? We unzip it, we now have single-stranded DNA, which is now susceptible to attack. So what ends up coming along to help out? You get these SSBs, your single-strand binding proteins, which will go through and bind to the single-stranded DNA to help stabilize it and prevent it from getting destroyed by some other enzyme. Okay. That then gets brought up to your polymerases. The polymerase has all sorts of different aspects to it. They've mentioned binding clamps. They've mentioned all sorts of other little pieces. So you can go through and specify and be responsible for all those pieces, or we can just say it's polymerase and it's doing all those fun things. Okay? And it ends up being what synthesizes outwards. Okay? You'll notice that on our lagging strand, we go through... Oh, they kind of crammed it in there. On our lagging strand, you'll notice that you do have the Okazaki fragment. You'll also notice that you'll see more of the primer molecule. Why do we not see the primer molecule on our newly synthesized or our leading strand? It's probably already replicated further out, so we don't see that. Okay? That primer strand is still necessary to initiate. It's just way past our reading frame, our visible frame right now.
You said a primer is just like the single strand?
Our primer is RNA, okay, which helps to initiate and start the synthesis process. Once it's started, we now start bringing in deoxynucleotides. Okay. Later on, something has to come back through and remove that RNA and then convert it into DNA and bring in the actual deoxynucleotides as opposed to the ribonucleotides. Okay, so lots and lots of repair then needs to happen after each of this. Okay. So errors in replication, we kind of talked about this. You get one mismatch in roughly 10 to the 9th to 10 to the 10th base pairs. If you're looking at that kind of a percent error, that's pretty insane. Okay? Which means we have to have some kind of proofreading mechanism to go through and remove those incorrect nucleotides when they do happen, because that can potentially lead to cell death, as we kind of addressed. Errors in hydrogen bonding also happens as well. So what we're talking about with the hydrogen bonding, which we did with the model in back, is that if we get two nucleotides that don't match exactly right, okay, the hydrogen bonding interaction does potentially line up and now allow it to potentially work, just not perfectly. That actually improves, or not improves, worsens your error. So 1 in every 10 to the 4th or 10 to the 5th base pairs as opposed to 10 to the 9th or 10 to the 10th. Okay? Because that hydrogen bonding interaction isn't really an error on the part of the enzyme. That's more the hydrogen bonding action actually kind of worked. So it's not really making an error. It's just synthesizing what it sees as being valid. As far as repair mechanisms, you get a whole bunch of different mechanisms. They all are sort of related to each other and sort of different. So we've got that list, which is how your textbook describes them as well. I don't find that particularly helpful, but you get different types. They do give you some nice pictures as far as proofreading and repair goes. So for instance, we had a mismatched nucleotide we're going to get a slight bump in our backbone. Okay? That bump can be detected by a repair enzyme. The repair enzyme can come along, find that place, and it makes a nick. Okay? That nick is ultimately saying what we're doing is breaking the backbone. We're breaking the phosphodiester backbone. Once that's now broken open, it will then re-replicate, or it needs to re-replicate, a large section of DNA, or at least that section. Okay? Since it can't quite figure out where to cut on the second end, it ends up doing this loop process. Okay? And it grabs another perfectly normal section, and it does another nick. Okay? That now enables that section of DNA to fall away from the structure. Because the only thing holding it there now are hydrogen bonds, which are your forces, no longer covalent bonds. That section can be removed, and now we have this giant gap of single-stranded DNA. This is where our repair enzyme can now come in and go through and re-replicate that section, and theoretically not make the same mistake. Once it's re-repaired that or put in the new nucleotides, again, you're going to need that ligase that they mentioned in the video to seal that phosphodiester backbone back up. Okay. We get a little bit of genetic freedom, so we can do some mutations, okay, almost intentional mutations. So if we encounter a new environment, we want to be able to mutate. So the whole point of mutations is to change our ability to respond to an external stimulus. Okay? So there is already some of this there. That's looking at these kind of same process or same idea of these nicks swapping DNA strands between different sections. This becomes more relevant, say, for your viruses that are trying to incorporate their DNA strands in, but we can do it to our standard stuff intentionally as well. Right? So this is your genetic recombination. You get the two different versions, either homologous or non-homologous, depending on where your information is coming from. When we move to eukaryotic replication as opposed to prokaryotic, as we already kind of said, it's a lot more complicated. The most studied one happens to be yeast, and you get your cell growth divided into all these different phases, all that other fun stuff. So you're getting a similar kind of process. You'll notice that all the symbols, okay, our alphabet soup of names has changed around a little bit. Ultimately, they're just trying to invent some name that sort of makes sense. So ORC, you're looking at an origin of replication. And you need different other proteins to come in to facilitate and cause this to break open. And that's what all of these fun little circles and drawings and colors are supposed to represent. A swarm of proteins and enzymes coming in to facilitate and start the process. Once it's started, you're going through a similar process with different polymerases, going through and processing and replicating your DNA. Your equivalent to polymerase 3 is known as PCNA. This is what your sliding clamp is. So it clamps around DNA and allows for the passage of DNA through it. So we get some kind of nice, pretty pictures of DNA through the center, passing through the center of our protein structure. You notice lots of alpha helices and all sorts of other fun stuff. So it's kind of pretty pictures, but that's about all we really want out of it. If we look at all our differences, you've got your polymerases, and then you've got your individual different types and segments of what each of those things function and do. You get slightly different names. Notice in the prokaryotes, we call them polymerase 1, 2, 3, and in eukaryotes, alpha, beta, delta, all that fun stuff, just so that we're trying to specify some difference between them. They are officially different, so we had to come up with some kind of name behind it. Okay? You'll notice that the Okazaki fragments does show up here. I know, I wasn't too far off. In our eukaryotes, we're looking at 150 to 200 residues. In your prokaryotes, you're looking at about 1,000 to 2,000 residues.
Which is counterintuitive.
How counterintuitive?
No, because of organisms and having more.
Yeah. The eukaryotes are more complex.
Right.
So,
but less of them.
And you're going to, I'm not even sure if this is true, but you might get potentially less mutations because they're more complex. So we might want fewer or shorter Okazaki fragments because that's less to process, or less possible mistakes. Again, how do the Okazaki fragments get made? I don't know that, okay? Which is a really, really good question because it's supposed to match that forming DNA strand or your lagging strand. Biggest difference between prokaryotic and eukaryotic? I don't remember. Oh. Well, we'll come back to that later. Okay, we were talking about what happens as we age. One of the things that happens as we age is this telomere section. So oh, that's what it is. I remember now. Anybody remember prokaryotic versus eukaryotic DNA?
Guessing it has to do with the telomerase. mRNA.
Circular DNA versus linear DNA is our big, big difference. So in the case of prokaryotes, there is no end, there is no beginning. Okay? So everything is kind of nice and tightly compacted within that circle. You open it up, you replicate, you're going to maintain that circle. What happens with eukaryotic DNA? Okay, well, in the eukaryotic DNA, it's linear, which means there is a beginning to it, and there is an end. Why does that become problematic? Well, if we look at, say, a bad enzyme, so here's a bad enzyme, we're just going to call it bad. Okay. If we're looking at circular DNA, there's no beginning, there's no end, everything is equally likely to get attacked. Okay, so we could compare this to the endonucleases, I think there were nucleases, and our exonucleases when we were looking at our carbohydrates. Which one was that? Amylase. Okay, if we think back to our amylase, sugar enzymes. Okay, the exonuclease could only attack on the outside, whereas the endonuclease could attack anywhere. And we notice the endonucleases tend to react faster because the relative concentration of sites that they can react at is greater because they can attack anywhere. So if we're looking at a bad enzyme, okay, everything within there is equally accessible. If we take a look at our other end, is everything equally accessible? No. These ends are now much more accessible to your bad enzyme, okay? Which means that end can be destroyed through time. Okay? So that becomes a bit problematic in the case of eukaryotes, because eventually what ends up happening? If this is now all my genetic information, okay, this allows me to live. If my bad enzyme comes through and deletes this section, I only have half of what I need to live, which means I'm dead. That kind of sucked. Okay? So we need to come up with some way to protect the genetic information. Okay? And since we know those ends are more susceptible to attack because they are now more open, we need to put something out there that we don't need. It's there just as a protection. That can be just a little protective cap. So what we end up doing is attaching on these extra strands of useless information. They don't store the information necessary to live, they just store nucleotides. Okay? So now if my bad enzyme comes along and it removes some of that section, I still have all of the core material that I need to live. Okay? So I still can produce all of the enzymes necessary for the Krebs cycle, for anything, to go through and continue. But eventually what happens over time, we've found, is that the bad enzymes do end up whittling away at this. And over time, that telomere, or that section of DNA, gets eroded until eventually we start eating into what we need to live. Okay? Which is our aging process. Okay? So there's a strong correlation between aging and that cap of just kind of BS DNA. That BS DNA needs to have a better name to it. That's where we come up with telomeres. So telomeres are just this extended cap of genetic, or not genetic information, but this extra piece of nucleotides at the end of our DNA. What that does is now when bad enzymes come through and start cutting away at those tips, we don't lose our genetic information and we continue to live. But what we've found is as we get older, those tips get smaller and smaller so that eventually the bad enzyme starts to cut into, or our degrading enzymes, start to cut into our genetic information. Okay? That starts to remove our ability to produce certain enzymes. Well, if I can't produce those enzymes, that process, that biochemical process stops. Okay? Then what we start doing is moving out of homeostasis and everything tries to compensate, okay? Which may mean less motion. Well, if you start moving less, predators can now pounce on you and you're dead. Okay? So there's all sorts of other things that can contribute to this. So what we ended up finding was that we have these long extended sections, our telomeres. Okay? And what we found even more interesting is that there's an enzyme called telomerase. Okay? And what it will do is bind to DNA, and it will attach these sections of nonsense DNA strands to the end of our genetic information. And those nonsense DNA strands will act as a protection, okay, so that we can continue to live. What we have found by studying DNA is that in very young people, those telomere strands are really, really long. In very old people, they are very, very short. Okay? Which would suggest at birth we can now predict how long you would potentially live based on the length of the telomere. Except as an interesting continuation of the study, they found that there are certain people that are very old that still have very long telomeres. And the question became, why? It turns out that as we get more stressed, we increase the concentration of our bad enzymes. What do the bad enzymes do? Eat away at the telomere. So if we live a very stressed life, we are now constantly exposed to these enzymes that are degrading our genetic information, which now slowly ages us, or increases the speed at which we age. Okay? Which is kind of depressing. They also found that you can change the length of your telomere by reducing your stress. So by reducing stress, you can then limit the presence of that degrading enzyme, and at the same time you give telomerase the opportunity to attach more nonsense DNA, extending the length of the telomere, extending the length of your life, due to health causes, at least. They don't stop bullets.
We found a correlation between, nationality and, I think the oldest person just died here at 100.
There are all sorts of studies along those lines, yeah, and I don't want to open that can of worms. But there's lots of nutrition, diet, alkaline water. I mean, you can get all sorts of different things explaining why someone lives longer than somebody else.
But ultimately, doesn't it really boil down to their DNA? I mean, because that impacts it hugely.
The DNA is ultimately what codes for all your genetic information, which is your proteins, the way you process foods, all of that stuff.
Right.
So there is a lot of information stashed within your DNA. It is a huge contributing factor to how long you live.
Right.
Is it the only factor?
No.
That's where things kind of become
No, but I
gray.
think that,
Yeah.
I mean, when you start talking about different, diseases and, susceptibility to diseases and stuff, it all boils down to genetic information, because you
Yes.
can be more or less susceptible to those stresses.
Exactly right. So that's kind of our end, I believe, of chapter 10. So it's our quick overview of replication.
So for those enzymes that go bad, is that controlled by the DNA, or is it just over
It's
time?
not that your enzymes go bad necessarily. It's that there are always enzymes present that help in the degradation of everything, because you need those pieces to do something else. So what happens if you get stressed? And we'll actually go ahead and stop the recording.